Today is the 11th August 2020, and it's my pleasure to introduce Andy Hill from Australia. Andy is a professor and head at the Trobe Institute of Molecular Science at Trobe University. His group studies neurodegenerative disorders such as Alzheimer, Parkinson's, and prion disease. So his lab has a major interest in the role that EV play in the pathophysiology of this disorder and their therapeutic and diagnostic potential. Andy established the Research Center for Extracellular Vesicles at Trobe University in 2018 and has served on the board of ISEF since 2012, including the past 4 years as president. So today I welcome Andy to present his lecture on extracellular vesicles, misfolded proteins, and neurodegenerative diseases.
All right, excellent. Well, hello everyone. It's great to see you all here this morning, or evening, or afternoon, wherever you are. And I'd like to thank Carolina, Dolores, and Jan for inviting me to come and give a talk today on extracellular vesicles and their role in neurodegenerative diseases. And what I thought I'd do today is talk about, give a bit of history to some of the work we did when we started working on EVs, and present a bit of unpublished data, but also some recently published work from the lab. And as Carolina mentioned, my lab's based at Trobe University, which is in the northern suburbs of Melbourne in Australia. And this is our building called LIMS, which is the Trobe Institute for Molecular Sciences. And we like to joke that these bits here are actually EVs budding off from the surface of the building. Right. So my lab's had a long-standing interest in neurodegenerative diseases, and many of these diseases are associated with proteins that misfold and deposit in the brain, particularly as we get older. And aging is one of the major risk factors for these neurodegenerative diseases. So diseases such as Alzheimer's disease are diseases that mainly affect our aging population. The diagnosis of these diseases is usually difficult, and it's because it's hard to diagnose a disease in the brain that is very slowly progressing, and it's usually only after clinical symptoms have occurred that you can start to define a definitive diagnosis of the disease. However, there's been some recent advances in brain imaging, which are helping to diagnose these diseases earlier, and just shown over here is some PET brain images of a healthy control and a patient with Alzheimer's disease. And both of these individuals have been injected with a tracer that picks up aggregated forms of a protein called amyloid beta that deposits in the brain of people with Alzheimer's disease. And as you can see in the healthy control, you don't see much staining, but in the patient with Alzheimer's disease, you can see a lot of staining for this misfolded protein, particularly throughout the cortical regions of the brain. So the diseases that I'm talking about today include things such as Alzheimer's and Parkinson's diseases, again, both diseases of our aging population. I'm also going to spend some time talking about some rarer forms of neurodegenerative diseases, such as the prion diseases. And there's also other diseases such as Huntington's disease, which is an inherited form of neurodegenerative diseases which occurs in families who inherit mutations in the Huntington gene. And also I'll talk a little bit today about amyotrophic lateral sclerosis or motor neuron disease. So these neurodegenerative diseases all have very distinct pathologies and symptoms, and they also are associated with different proteins that deposit in the brain. So for example, Alzheimer's disease, the proteins that deposit and form these plaques in the brain are called amyloid beta and tau. And again, here is the example showing the amyloid beta staining in the brain. In Parkinson's disease, it's a protein called alpha-synuclein, and that deposits in a very specific brain region called the substantia nigra. Prion diseases, it's the prion protein that misfolds. And in amyotrophic lateral sclerosis, it's a variety of proteins. They tend to be RNA-binding proteins, such as TDP43 and Fuzz, in addition to superoxide dismutase 1. And there are many different mutations in these genes that can cause ALS. And so, this is a classical picture showing the degeneration that can occur in these neurodegenerative diseases. So on this side here, you can see a healthy aged brain and one from a patient who suffered from advanced Alzheimer's. And you can see the massive loss of brain tissue during this advanced case of Alzheimer's disease. So a common feature of these diseases is not only the misfolding of these proteins, but it's actually the spread of these misfolded proteins throughout the brain. So the misfolding tends to start in one particular brain region and then over a long period of time spreads to other brain regions where you see the pathology spreading. And so we think EVs might play a role in spreading these misfolded proteins throughout the brain and being involved in these diseases. So just to start off with, I'll talk a little bit about the role that EVs play in the nervous system. And as you know, there are several different cell types that are associated with the nervous system. These obviously include neurons, but also cell types such as oligodendrocytes, microglia, and astrocytes. And a lot of work has shown that EVs released by these different cell types are involved in maintaining the nervous system and are involved in quite specific processes. So an example being the release of EVs in response to inflammatory conditions by the microglia and astrocytes. And astrocytes can also release EVs that are neuroprotective. They can help to protect neurons. And so there's various different functions ascribed to EVs that are released by these different neuronal cell types. And also that there are various different stimulators of EV release, such as the release of glutamate by neurons can induce the release of EVs from oligodendrocytes, and other things such as ATP can again induce the release of EVs from microglia and also astrocytes. So one thing that's also of note is that EVs that exist in the brain that are released by these different cell types can be released into the periphery, either by crossing the blood-brain barrier into the circulation where they can enter the bloodstream. And it's been shown that EVs can cross in both directions, from the blood into the brain and also from the brain into the circulatory system. So as we'll talk about a bit later in the presentation, we think that looking at EVs in the circulatory system might give us a window into diagnosing diseases of the brain. Also, the brain can release EVs into the cerebral spinal fluid, and this is another biofluid in which EVs derived from the brain can be isolated and have diagnostic potential. So on the first slide, I showed you many of those proteins that are involved in neurodegenerative diseases. And over the last 15 years or so, many studies have shown that a lot of these proteins involved in neurodegenerative diseases are actually found associated with extracellular vesicles. This includes, and it's not limited to, proteins such as alpha-synuclein, which has been detected in both its aggregated and its normal monomeric form in EVs, and alpha-synuclein is involved in Parkinson's disease. You can detect the normal and the aggregated form of proteins involved in amyotrophic lateral sclerosis or motor neuron disease. This includes superoxide dismutase 1, but as I'll show you later, also TDP43 and Fuzz and other proteins. In Alzheimer's disease, the amyloid beta peptide, its precursor, the amyloid precursor protein, and also tau can be found in its normal and disease-associated forms associated with EVs. And also, as I'll talk about in a moment, the two forms of the prion protein, the normal form and the disease-associated misfolded form, can be found on the outside of EVs. So as you all know, EVs don't only contain proteins, but they contain lipids and nucleic acids. And also, as I'll show you later, we find some disease-specific microRNAs associating with some of these different diseases. So I'll start by telling you how we entered the EV field, and that was through our study looking at prions, which are proteinaceous infectious particles that cause neurodegenerative diseases that are classified, that are called generally prion diseases. And they include diseases such as Creutzfeldt-Jakob disease, or CJD, or bovine spongiform encephalopathy, which is otherwise known as mad cow disease. And so this is a really interesting infectious agent. It's hypothesized to be protein only in nature, so there's no nucleic acid component of the infectious agent. And really the whole premise around the prion hypothesis is that you have a normal form of the prion protein, which is called PRPC, where the C stands for cellular. And this is a protein we all express in our central nervous system. And it's a protein that is attached. So we've got here these gray bubbles here. This is the cell surface. We have what's called a GPI anchor shown here. And then this normal protein, PRPC, is this alpha helical rich protein with two of these N-link glycosylation sites that are flopping around in the wind there. And it has a long N-terminal unstructured domain. Now, what happens in the disease process is that this protein changes its structure from being rich in alpha helices to being rich in beta sheet, and that causes it to aggregate and deposit in the brain. And what happens is when you form this aggregated protein, the conformation has changed from one that's rich in alpha helices to one that's rich in beta sheet, and it becomes known as PRPSC. And SC stands for scrapie, which was one of the first prion diseases to be described in sheep. And so this protein then, this beta sheet rich protein, can then go and recruit more copies of the normal form of the protein and convert them into this bad beta sheet rich form of the protein. And this becomes this infectious agent that can recruit more copies of the normal protein and convert it into this disease associated form. And as I'll show you, we think that EVs play a role in carrying this abnormal form of the protein and converting the normal form of the protein into more copies of the bad form of the protein. So prions are spread by the post-translational conversion from this alpha helical rich PRPC to beta sheet rich PRPSC. And since the prion hypothesis was proposed in the early 1980s, it's been shown that there are lots of prion-like mechanisms in other neurodegenerative diseases. And that is to say that aggregated proteins such as A-beta or alpha-synuclein can actually seed the conversion of the monomeric form of the protein into more copies of the abnormal form. So there are several different mechanisms of how this infectious beta sheet rich protein can spread between cells. So when you have a prion-infected cell, you have the normal form of the prion protein, GPI anchored to the cell surface, and you also have the abnormal misfolded beta sheet rich form, PRPSC. This is the prion infectious agent on the surface of the cell. There are ways that this misfolded protein can spread between cells and start that chain reaction of converting the normal PRPC. And this can be through processes such as tunneling nanotubes, which have been shown to carry both the normal and abnormal form of the prion protein between two cells that are in very close proximity. So these nanotubes form when two cells are in close proximity, and they form these protrusions that connect one cell to another and allow them to share their contents. The other is through direct cell-to-cell contact, and this is where a PRPSC molecule in one cell connects to a PRPC molecule on a recipient cell and induces that conversion. So this cell will then become infected, have this beta sheet rich form of the prion protein on it. So direct cell-to-cell contact can also be a way of converting and spreading this misfolded infectious protein. And obviously, the mechanism that we're most focused on is the role of EVs, in particular small EVs or exosomes, in carrying the abnormal prion protein from cell to cell. And as I'll show you some data that shows that EVs are quite efficient at carrying these misfolded proteins that can spread between prion-infected cells and recipient cells, and also potentially into the bloodstream. So this slide summarizes about 10 years of work where we started to look at the role of EVs in spreading prion infectivity. And we started off, and this is work that Laura Vella started off our EV projects in the lab when she was doing her PhD, used a cultured cell system where we had cultured cells that expressed the normal form of the prion protein, and we exposed them to some of that beta sheet rich form of the prion protein that we'd obtained from some mice that had died of prion disease. What happened in this case is that the cells take up the infections, they take up that bad form of the prion protein, and as we passage the cells for several weeks, we start to see the infection spreading amongst the cells in culture. And we use a biochemical assay to test for the presence of the beta sheet rich form, the aggregated form of the prion protein, and that involves treating the cell lysate with a protease that digests away all of the normal prion protein. So this is before proteinase K treatment, and this is after, and we can see we've got rid of all of the prion protein immunoreactivity. And in cells that are infected with a prion strain that we call MUO2, you can see that after proteinase K digestion, we've got these protease-resistant bands that represent that beta sheet-rich form of the prion protein. What Laura did was she isolated EVs from these prion-infected cells and injected them along with cell lysates from the same cells into susceptible animals. And we showed that the exosomes from 2 different cell lines infected with prions caused a clinical prion disease in mice at a very similar time frame, incubation period, to if we injected the cell lysate from these cells. To look at whether we needed cell-to-cell contact to spread the prion infection between cells, we also did a transwell experiment where we cultured prion-infected cells above a transwell membrane, which had a 0.4 micron pore size. And this allows EVs to transfuse across the membrane and interact with some non-infected reporter cells in the bottom of the dish. We use what's called a cell blot assay, which is where we basically grow the cells on a cover slip and then probe for the presence of this protease-resistant protein. So you can see here in mock-infected cells, we don't see any protease-resistant protein. But when we had prion-infected cells on top of this transwell and measured whether there was any infectivity in these cells at the bottom of the dish, we could detect that prions were able to transfer, diffuse across the membrane and infect these reporter cells. And this will become important for the next slide I show you. Another experiment that Laura performed was to take culture media from these, conditioned culture media from these prion-infected cells and use that to infect naive cells. And we showed that that was very effective at causing the uptake of infection. And when you isolate the exosomes from that culture media, that was even more effective at transmitting the infection, and that there's very little infectivity present in the exosome-depleted culture media. So collectively, this is showing that EVs are very efficient at transferring that infectious prion protein from cell to cell. Belinda Guo, who's a PhD student in the lab, she did some experiments where she looked at modifying the amount of EVs released by cells to seeing how that affected the spread of the prion infectivity from these infected cells here in the transwell to these non-infected reporter cells. And she used a couple of different approaches that I'll show you here. The first was to use a neutral sphingomyelinase inhibitor, GW4869. And in this publication, she also reports using siRNA against this gene as well. And we can see that when you treat these cells with this inhibitor, you get a reduction in the number of EVs released by the cells. You get about a 50% reduction. And when we measured the amount of infectivity that could be spread from the cells, infected cells in the transwell to the ones at the bottom of the dish, we found that with no drug added, we got efficient transfer of infectivity. But when we added the neutral sphingomyelinase inhibitor, which reduced the amount of EVs, we got a great reduction in the amount of infection that could be detected in these cells. So this was sort of proof of principle that reducing the amount of EVs released by these cells reduces the amount of infectivity transfer between the infected and the non-infected reporter cells. She then used another drug called Minensin, which has been shown to increase the amount of EVs released by cells. And you can see here in a dose-dependent manner, as you increase the concentration of Minensin, you get an increase in the number of EVs released by the cells. And when we did the same experiment here, where we treated these prion-infected cells with Minensin and measured how much infectivity could be picked up by the reporter cells, we saw a great increase in the amount of infectivity in a dose-dependent manner. So increasing EV release increases the amount of infectivity, decreasing it decreases the amount that you can detect. This is some work that Brad Coleman did in the lab as a PhD student that we haven't yet published, but we tried to look at what do we have to do to these EVs to reduce the amount of infectivity. So this was really some really nice biochemical work where we estimated the amounts of infectivity carried by these prion-containing EVs. And by performing several different biochemical treatments on the EVs, we tried to look at how we could modify the amount of infectivity. And so what we did was we took a sort of virological approach by doing a series of dilutions of the number of EVs we were adding to the cell, or the amount of cell lysate, which was our control. And I'm going to show you several graphs here, and what I want you to see is that when you see the graph move towards the left, it means that we have increasing infectivity, because as you've diluted the inoculum, you've got an increase in the signal of the protease-resistant prion protein, which I'm showing here. So the first thing we did was we asked the question, are the cell lysates or the exosomes from those infected cells more infectious when we control for the same amount of the protease-resistant beta-sheet richer form of the prion protein? So here we're showing this prion, the bad form of the prion protein in the cells, and also here in the exosomes. And we can see we've got a very equal loading of those two amounts of the infectious form of the prion protein. When we did a dose dilution of these amounts of the infectious form of the prion protein, we found that the graph for the exosome, the EVs, had moved to the left, showing that the exosome fractions are more infectious than the total cell extracts. And that this increased infectivity is not simply due to there being any more of this PRPSC in the EVs, because we've actually controlled for the levels here. So this showed that EVs and exosomes are a very efficient mechanism for transferring this infectious protein. We then thought, well, is there something on the cell surface that enables these EVs to be very readily transfers of this infectivity? So what we did was we treated the EVs with proteinase K, and we basically shaved off all the proteins from the outside of the EVs. And this just left those protease-resistant forms of the prion protein. And we did some transmission electron microscopy just to show that we hadn't altered the ultrastructure of the EVs using this treatment. And here you can see, comparing the EVs before the protease treatment and after the protease treatment, that there was no significant difference in the infectivity titer. So the transmission electron microscopy showed that there was no structural or morphological changes after the protease treatment, and that there's no exosome-related infectivity loss when you've lost all of these protein cofactors. So then we started to see, well, what happens if we break open these EVs that contain this infectious protein? So we lysed the EVs with hypertonic shock and sonication. And we did this where we aim to maintain intact membrane fragments of the EVs, but rupture the whole exosome. So just break them open into small membrane fragments. And again, we used electron microscopy. These are the EVs before the sonication and hypertonic shock. And afterwards, you can see we've broken them up into small fragments. And the bottom line here is this didn't affect the infectivity either. So the exosomal infection efficiency is actually enhanced by a membrane component. And it was only when we used chemical methods to break open and solubilize those membrane fragments that we saw a loss of infectivity. So again, we've got the control EVs here. We treated them with methanol, and then we also treated them with Sarcosil, and we can see that the methanol treatment, this is the control in red here before the treatment, and we can see the graphs now moving to the right. So after the methanol treatment, we've lost about half of the infectivity, and then we also treated with Sarcosil, we lost virtually all of the prion infectivity. So this shows that we're losing the lipid or membrane component, and that we think that it's a combination of the lipid and membrane environment in which that PRPSC is bound actually is the infectious agent. So now I'm going to move on to talk a bit about how we've used EVs in trying to diagnose some of these neurodegenerative diseases. And as you're all aware, EVs are implicated in a number of disease settings, such as cancer, neurological diseases, as we're talking about today, but also cardiovascular and other infectious diseases. And as we know, there's a great potential to use various EV cargoes as biomarkers for disease, and this can include RNA, protein, or lipids. But I think in developing EV-based diagnosis, there's a few technical barriers that we have to overcome, which include methodologies used to isolate the EVs, and also the sensitivity of downstream assays. I think those of us who work on EVs know that it's hard to get enough material to do a lot of in-depth work. And also another key technical barrier, which isn't limited to EV studies, but also more generally to biomarker studies, is that standardization of methodologies and validation of biomarkers is also a very important step. So this is our typical EV transcriptomic workflow that we use for identifying RNA-based biomarkers for various diseases. And we start with either cell culture supernatant, biofluids, or tissues. I'll show you some work we've done using brain tissues. We isolate the EVs, extract the RNA, do quality assessments, and prepare libraries for sequencing. And in our lab, we generally use small benchtop sequences, such as the Ion Torrent S5, which allow us to actually do the sequencing in our own lab and control all of the variables for doing the sequencing. And these small benchtop sequences are actually really good for looking at small RNA populations. And then once we've done the sequencing, there's a lot of bioinformatics that are involved in analyzing the data you get from the sequencer. So one experiment we did do is to look at, does the profile of microRNAs or RNAs in general, does that need you to isolate highly purified EVs? So we did an experiment where we compared the standard 100,000 K differential ultracentrifugation protocol to generate a crude EV pellet. And then we took this pellet and we floated it on an OptiPrep velocity gradient. And by this, we can get very highly purified populations of exosomes. Fractions 8 and 9 from our gradients contain the very highly purified small EV population. And what we did was we did some small RNA profiling using our sequencing method to compare the RNA profiles of the cells, the crude EV prep, and then the highly purified preparation through the OptiPrep velocity gradient. And to cut a long story short, what we found was that, as has been seen in many studies, the microRNA profile of the cells is quite distinct from that you find in the EVs. And what we found using this PCA plot here is that fractions 8 and 9, which contain our highly purified EVs on that OptiPrep velocity gradient, they sat in the same region as the graph as the crude EV prep, the ultracentrifuge prep. So this showed us that you didn't need to go to great lengths to get highly purified EV populations to actually get very similar RNA data. And fractions 7 and 10 are just the two surrounding fractions on the OptiPrep velocity gradient where you can see the RNA profiles do start to change from those where the EVs were found. And also we had very good overlap in the microRNAs that we could detect between the ultracentrifuged pellet and the OptiPrep velocity gradient fractions 8 and 9. So this was the first study published back in 2012 where we used next generation deep sequencing. So we used this now defunct sequencing platform. This is the solid sequencing platform, where we compared the EV profile in the exosomes isolated from control and prion infected cells. So we did the sequencing, we found some microRNAs that had differential changes, and then we validated it using two different qPCR assays. And we found a panel of 9 microRNAs that had differential expression levels in the prion-infected EVs compared to the control cell EVs. So this started us asking the question, can we use these exosomal microRNA profiles to diagnose neurodegenerative diseases? And this is the results of a study we published a few years ago, where we'd used samples that had been supplied to us by the Australian Imaging and Biomarker Lifestyle Study of Aging, which has been running in Australia for over 10 years now. What we did was we isolated EVs from the serum of 23 healthy controls and 23 Alzheimer's individuals. And using the sequencing approach, we found a panel of 16 microRNAs that were differentially expressed in the Alzheimer's disease participants compared to the healthy controls. So this was our discovery phase. We then took these 16 microRNAs and used them in a validation phase. And here we had access to 50 samples that we analyzed blind to diagnosis. But all of the patients had been diagnosed by standardized criteria for Alzheimer's disease, and all of them had had PET brain imaging for the amyloid beta, as I showed you on that first slide, performed. When we uncoded we performed the analysis using qPCR against these 16 microRNAs found in serum-derived EVs. When we broke open the code, we were told that we had 35 healthy control participants and 15 Alzheimer's disease participants in this validation study. So with the Alzheimer's cases, there are 15, and we're able to correctly diagnose using this qPCR assay on the serum-derived EVs. We correctly diagnosed 13 patients, which gives us a sensitivity of 87%. And of the 35 healthy controls, we correctly diagnosed 27 patients, giving us a specificity of 77%. And this isn't too bad for a blood-based test for Alzheimer's disease, which is otherwise quite hard to diagnose. But one thing of interest is that in 5 of these patients that we'd diagnosed, they had quite a high level of amyloid beta in the brain. And so if these represented cases of preclinical Alzheimer's disease, we think our specificity is going to go over 90%. And we're currently wrapping up a study now where we've been looking at a longitudinal study of cases from the ABLE cohort, where we're actually trying to measure these, whether we can detect these preclinical cases or not. But doing this study on blood also highlighted the fact that blood biomarkers might not actually represent what is going on in the brain. So we started off a long-standing project, and it's a great collaboration we have with Laura Vella at the Trobe Institute in Melbourne, where we've been trying to look at brain-derived EVs by isolating them from brain tissue and comparing them with the profiles we can detect in the blood. And this is to try and identify what are the brain-derived, what are the brain-specific microRNA species involved in disease? Can we capture them in the blood and identify a preclinical microRNA signature for these diseases? So key to these studies is a protocol that was developed by Laura, where she isolated EVs from brain tissue, from frozen human brain tissue, which underwent some centrifugation spins. And this is the key part of the protocol, which is this triple sucrose cushion. And this is where you can separate different sized vesicles isolated from brain tissue that's been treated with very mild protease and phosphatase inhibitors and endopeptidases. And we get a fraction called fraction 2 that contains what we think are brain-derived exosomes. They met all the criteria of the 2014 MICID guidelines, because we published this in 2017. And here you can see some EM of these brain-derived EVs. We can see that fraction 2, which is our EV-rich fraction, doesn't contain what were known as non-exosome markers, and they're rich in what are well-used EV markers such as syntonin, TSG101, flotilin-1, and CD81. We did some shotgun proteomics on these EVs, and we found that they contain many of the top 25 exosomal proteins according to ExoCarta, but more crucially, by mass spectrometry, we detected all of the proteins that we're interested in that are involved in neurodegenerative diseases, and this has all been published. Leslie's just published a recent paper in JEV where we've been comparing the EV microRNA profiles of serum with whole brain and brain-derived EVs. And this is to get that idea of, can we develop a liquid biopsy of the brain by looking for brain-derived microRNA signatures in the blood. When we look at the brain-derived EV RNA species that can be found, which is in that fraction 2, and we also look at fraction 3, which does contain some EV material, and compare it with total brain tissue, we can see a whole suite of different non-coding microRNA species. I don't have time to go through this today, but this has been recently published. What Leslie found by doing this microRNA analysis on the brain-derived EVs and the serum, and also the brain homogenate, the total brain from which the EVs were derived, we found some really interesting things. We found several microRNAs that have had the same microRNA expression level in both the brain homogenate and the EVs. We found some, and this has been seen in other biofluid studies, where we see some microRNAs that are upregulated in the brain-derived EVs, but downregulated in the brain tissue from which they were derived, and also ones in the opposite direction as well. When we look at the brain-derived EVs and compare them with the serum-derived EVs, again, we see a similar picture. And we're starting to build up a map of microRNAs that either stay the same between the brain and the periphery, or go up in one and down in the other, or vice versa. And so we're starting to build this brain-derived EV map that we can detect in the periphery. Now, just coming back to the prion diseases, so I showed you that data where we'd shown the microRNA profiles differed between control and prion-infected cell cultures. And we've now taken that into an animal model, and I'll show you some data from some human samples as well. And our animal model involves injecting animals with infectious prions, and it takes them about 20 weeks to develop the terminal stage of clinical prion disease. And I'll show you some data from 3 different time points of this 20-week clinical study in the animals. So we look at animals 3 weeks after inoculation with prions, and this is the preclinical phase, so we don't see any pathology in the brain, and we don't see any of the misfolded form of the prion protein when we look at it on a Western blot. At week 13, we're starting to see some mild pathological changes. So a characteristic of prion disease is the appearance of these spongy holes in the brain. So you're starting to see some of those appear. And at week 13, we can start to see the appearance of the misfolded form of the prion protein. So this is the bridge between the preclinical phase and the clinical phase. And then in the terminal stage of the disease, you can see there's a lot more spongiform change in the brain, and the brain is filled with this misfolded form of the prion protein. And clearly this is the clinical phase of the disease. So we started by wanting to track the expression of expression levels of microRNAs and EVs isolated from these prion-infected mice. So we had 8 to 10 mice per group, per time point, so it was a very large study. We extracted the thalamus, which is one of the key regions involved in the disease. We've also been looking at the brain-derived EVs, but for the samples I'll show you today, we've been looking at the expression levels of microRNAs in the serum-derived EVs. This is really to see, could we use this as a prototype for a diagnostic test for these diseases? So this shows you some of the data we got from looking in the thalamus. So this isn't looking at EVs, this is looking at that whole brain tissue where we looked at the microRNA levels in this highly affected region of the brain. And we looked, I'm just showing you the data here from week 3, 13, which is that bridge between the clinical, preclinical and clinical phases and the terminal phases of disease. And the circus plot shows in the outer ring here, this is week 3, in the middle ring here, it's week 13, and T is the terminal phase of disease. And each one of these gray bars is an individual microRNA. And we can see by doing this longitudinal study, we could find some microRNAs that started off with very low levels of expression at that preclinical phase. The expression went higher at that midpoint between the preclinical and clinical phase, and then decreased expression in the terminal phase. This microRNA here was really interesting because 3 weeks after inoculation with the prions, we found very high levels of expression, and these decreased to the terminal stages of disease. And as you look across the whole course, you can see a combination of different microRNAs that go up, down. And so we've got some nice preclinical markers, some mid point of the disease markers, and some terminal disease markers. When we looked in the serum EVs extracted from these mice that were infected or not with the prions, we saw a very similar picture, and we see some microRNAs that start off with high levels of expression in the preclinical phase that reduce in the terminal phase, and vice versa. So we think this is a really good model to show which particular microRNAs are involved in the pathogenesis of prion disease that could actually be used as disease biomarkers. And in a study where we collaborated with a group in Germany that had been collecting human prion disease samples, we used this panel of microRNAs we'd identified from the mice, and we got very good sensitivity and specificity looking again at serum-derived EVs. So we think that these microRNAs might be a really good diagnostic marker for prion disease. So now I'll just finish off with some recently published data in the few minutes I've got left, where Natasha Vasilev, and again this is a collaboration with Laura Vella, and Leslie Cheng, has been looking at the isolation and characterization of brain-derived EVs from amyotrophic lateral sclerosis or motor neuron disease brain tissue, specifically in the motor cortex. And Natasha's used the same ultracentrifugation protocol with the sucrose gradient for isolating these tissue-derived EVs, and she showed the 3 fractions here, with fraction 2 showing that they morphologically resemble small EVs, and we've got some nanocyte traces here. She's also showed in characterizing these that the fraction 2 vesicles contain the small EV markers TSG101, flotillin, and syntonin, an absence of calnexin, VIP, and RPL5. Natasha was interested in looking at the protein changes between control and ALS tissues. And she found that the brain-derived EVs isolated from the ALS brain tissue were enriched in TDP43 and found an interesting result that C-terminal fragments of TDP43 were significantly increased in the brain-derived EVs from the ALS samples compared to controls. She then undertook an in-depth proteomic analysis comparing the EVs from the normal control and the ALS tissue and also the total brain. And she showed that there are proteins common between the brain-derived EVs and the total brain tissue, but there are different relative abundances between the brain tissue and the EVs, which again has been seen in many different studies. And she's able to detect over 1,000 proteins in both the EVs and the total brain. So she had really, really good coverage here. But the key thing was to look at what are the differences in the EV protein profiles between the control and the ALS samples. And here she found 16 proteins that were deregulated in the ALS samples by comparing the EV protein profiles between the ALS and the control samples. And several of these proteins that she analyzed using this unbiased screen have been shown to have direct links with ALS pathology. So it's great to be able to pull out these proteins in EVs that have already been shown to have links with ALS pathology. And so these proteins all exhibit RNA binding activity. So as I mentioned earlier on, a lot of these proteins involved in ALS are RNA binding proteins. And so we think this might show some biomarkers that could be useful for the diagnosis of ALS. And so now I'll just finish off by concluding that I've shown you that the structure of whole intact exosomes is important for ensuring the efficient transmission of prion infectivity. It was only when we broke open the vesicles and solubilized the membranes that we were able to abolish the spread of the infectivity from cell to cell. And that deep sequencing of EV RNAs from prion-infected cells, the brain and serum of mouse models, reveals specific microRNA signatures that have diagnostic potential. And I didn't show you much of this data today, but a lot of the microRNAs we're pulling out, when we look at the targets of these microRNAs, they actually have very well-formed links with disease pathways. So in the lab, we're looking at the functional aspects of some of these microRNAs and how they actually might be involved in the disease. And finally, that EV microRNA signatures are associated with Alzheimer's and prion diseases using clinical samples. And we're currently doing a number of studies using a larger number of samples, and including a study on Parkinson's disease I haven't been able to talk about today, where we think that EV microRNAs might be very useful in the diagnosis of these neurodegenerative diseases. So with that, I'd like to thank you all for listening, and thank you to the many people that have been involved in this work over many years, especially Laura Vella, who started the EV work in my lab, and we continue to have a great and productive collaboration. Leslie Cheng has been doing all of the microRNA work in the lab. I spoke about Natasha's work, so I'd like to thank all of the lab past and present for their contributions to this work. All of our collaborators as well, particularly Vicky Lawson and Laura Ellett at the University of Melbourne for our long-standing collaboration on the prion work. And I'd like to thank all of our funders, and thank you all for listening, and I'm happy to take any questions.
Thanks, Andy, for this wonderful presentation. Very interesting. I do have some questions, but I want to go through the questions in the chat box first and ask everybody when they ask the question to mention where they're coming from, which institution and geographical area. So Phil has a lot of questions. Phil, maybe we can start with a couple of them and then switch to somebody else. So, because it's like 6 questions or something like that. You're muted.
Andy, this is an overwhelming amount of work with very, very difficult material. This is fantastic stuff. Are the involved in these special? Do they have special lipids in the membrane? Are they a subpopulation or a sub-subpopulation? And what is the relationship between the special microRNAs and the folded proteins? That's enough.
Okay, thanks Phil for your questions. So I didn't show it today, but we did do some studies where we'd use that OptiPrep velocity gradient to try and really look at what kind of EV is carrying this infectious protein. And when we compared the fractions from the gradients between control and prion infected cells, we found that using cryo-electron microscopy, we could detect different membrane topologies that associated with cells that were prion infected and those that weren't. It's been very difficult to pull that apart even further. And one thing we did do was we did do some mass spectrometry trying to look at the lipid profiles of these different populations, but we didn't find a lipid species that really reliably distinguished between the populations from the control and the prion infected cells. And I think that's something we have to remember. We're always looking at a population of vesicles, and the more you look at them, the more heterogeneous they are. So yeah, that's until we can get down to single EV kind of science, I think it's going to be hard to pull that apart. What was your next question? Sorry.
What's the relationship between the special microRNAs and the misfolded proteins?
That's a good question too. I mean, the microRNAs are more of a marker, so they might not have a direct relationship with the proteins. What we're seeing is some of the microRNAs, when you look at the targets of those microRNAs, they're usually processes involved in the disease in general, either sometimes in processing those proteins or other effects of the disease. So the link isn't always direct between the microRNA and the protein.
Last question, is misfolding a normal process, or are there normal processes that depend on misfolding?
So I'd say, I mean, given protein folding is you know, when you go back to university lectures on protein chemistry, and I remember seeing those folding funnels of, you know, the challenges of how do you fold, you know, a polypeptide chain into a three-dimensional structure. And I think misfolding is probably happening all of the time. And I think these diseases of aging are probably more prone to the protein misfolding pathologies because the cells probably lose their ability to deal with that misfolding. So clearing misfolded proteins is a natural process. And I think when you lose that ability to clear those proteins, I think that's when it becomes a problem. So I think misfolding occurs all of the time. And, you know, there are things that can promote that, such as a lot of these diseases, mutations in the genes that encode these proteins predispose to the disease. So subtle changes to the proteins can promote that misfolding. So I think misfolding is happening all of the time.
Thank you very much.
Thanks, Phil.
A quick follow-up to this question. So if misfolding happens all the time, are the misfolded proteins always taken by the endosomal machinery to lysosomes or eventually shed into extracellular vesicles?
Yeah, that's a great question. And I think, you know, coming back to, you know, some of the first proposed roles for exosomes was the way that cells get rid of junk. And so you could proposition that this could be the way that the cell gets rid of misfolded proteins by shedding them through EVs. yeah, the lysosomal system is involved in breaking down these misfolded proteins as well. They're very hard to study. It's hard to study protein misfolding in the cell as it actually happens. And, there's some very nice tools for doing this using different fluorescently labeled proteins, but it's actually quite hard because you're looking at one misfolded protein in a whole sea of other forms of the same protein. So it's very hard to look at.
Let me ask you another question that I had ready also. Do you think these misfolding proteins are only released in exosomes? So why do you think this is so specific for exosomes and not for other vesicles?
I think some of the misfolded proteins can be secreted from the cells as well, or in the case of prions, sometimes breaking that GPI anchor releases it, so it doesn't necessarily have to be EV related. And I think sometimes these proteins might be EV related because they've been released by the cell through another mechanism, and they've just stochastically bound to an EV and can be associated that way. So again, I think there's a whole lot of other mechanisms. So I don't think EVs are the sole way. I think that slide I showed with the different mechanisms of how prions can spread from cell to cell, I think there are overlapping mechanisms.
Okay. Okay, there is a question from Kalyani Patil. And Kalyani, can you please say where you are located?
Yes, so I'm here in Qatar. We just moved in November and the corona started, so I'm not associated with any institute. Thank you, Andrew, for the talk, and I really appreciate your work. So I have a question which is in 3 parts. So the first is, since age is a common factor in all the neurodegenerative diseases, so can these diseases coexist? And if yes, are there any misfolded proteins that are shared between any 2 neurodegenerative diseases? And the third is that if they share any proteins together, can EVs shift the balance towards that disease which shows higher level of the misfolded protein expression?
Okay, so I think the coexistence of neurodegenerative diseases is a really interesting question. And I think you can only really look at that pathologically by looking in the brain for these different proteins after someone has died of the disease, because I think the clinical presentations of these disorders are quite mixed as well. There have been some cases, and they're not very common, of coexisting pathology, say with prion disease and potential Alzheimer's disease. But the clinical presentation would be quite different. There are some shared misfolded proteins. So tau, for instance, is involved in Alzheimer's disease, where it forms these neurofibrillary tangles, and those together with amyloid beta plaques, and there's a lot of debate as to which one is more important. So those are involved in Alzheimer's disease, but tau itself can cause different forms of neurodegenerative diseases. There are families with mutations in tau, for instance. So, and actually with the prion diseases, you've got one protein, different mutations in that protein will give you very, very different diseases. So you can get a single point mutation in PRP can give you a disease called fatal familial insomnia, which is very different to CJD. So yeah, there's shared misfolded proteins, but even the same protein can have a very different phenotypic effect in the disease. Sorry, I forgot your last question.
Yeah, so if any 2 diseases share the misfolded protein, so of course they would be shared in the extracellular vesicles. So how do extracellular vesicles decide how they decide their bias towards one particular disease?
Wow, what a great question. I think that would be quite, I'm not sure how we could look at that, because again, when we're looking at the EVs, we're looking at a population, and no 2 EVs are going to have the same complement of proteins and things on them. So I think it would be a really rare event. I'm not sure if you'd pick it up that you might have the two misfolded proteins on the one EV, for instance, and how would that influence what happens in the recipient cell? But no, it's definitely something to think about. It's a great question. Thank you.
Thank you. Thank you. I was just thinking because it might shift balance towards a disease that shows higher expression of that particular misfolded protein. So
Sure.
if the EVs has, say, a protein A that shows higher expression, then it might shift balance towards that particular disease.
Sure. Okay. Excellent. Thank you.
Thank you.
So then we have a question from Alisa Weaver from Vanderbilt University in Nashville.
Good. Thanks, Dolores. You introduced my affiliation. So, Andy, that was a great talk. And I've always found the prion story fascinating with respect to extracellular vesicles. My question is not that different from one that Phil asked. Actually, I was wondering why, you know, exosomes or small extracellular vesicles are so much more efficient than your cell extracts at converting the cellular form to the scrapie form. And I had been wondering if the lipids, you know, might play a role in that, because you digested away all the other proteins and still it was, you know, just as efficient.
Yeah, I think, you know, it's been very hard for people to recreate an infectious prion from highly purified components. And by that I mean, if you take a recombinant prion protein, you can misfold it into a beta sheet form, but it's not necessarily infectious. And it's only when you add back things, and mainly they're sort of charged molecules such as RNA and lipids, you can actually recreate that infectivity. So I think the EV presents a really, just the perfect vehicle in terms of, you know, it's got a restricted number of proteins and other molecules. It's got this misfolded protein in a lipid environment. We don't know what that is yet, but that seems to be the key thing for the recipient cell to take up and to start that replicative process of converting the normal proteins into the bad form. I think in the cell lysates, you've got a whole soup of things. It's still infectious, but not as efficient as when you've got this thing packaged in the vesicle.
And you think that the RNA is a part of it as well in terms of the infectivity, or is it more just a biomarker?
Look, I'm really interested in the idea that small RNAs might play a role in the infectivity. There's a whole sort of field within the prion area looking at different strains. So the same protein misfolded in different ways can adopt very different pathologies. And that could be, you know, the early work looking for a genome associated with the infectious agent didn't detect any nucleic acids except for ones that were less than 50 base pairs in size. And of course, now we know about small RNAs, it's possible that they could play a role in mediating the infectivity. But it's a very hard system to work with. And again, we're working with these misfolded proteins that are sticky, they bind to all sorts of things and hard to purify, notwithstanding all the biosafety aspects of it as well.
Interesting.
Okay, thank you. Next question from Meruyert Imhanbekova. Are you there?
Yeah, I'm here. Hi, thanks, Dr. Hill, for great talk. And I'm from McGill University, Montreal. So I have a couple of questions regarding the form of the protein, misfolded protein that you identified in EVs. So was it the beta sheet form, and did you specify the location of that protein within EVs? Because on your first few slides, you showed that some proteins, such as a beta, is located more on the membrane, and some proteins within the EVs. And has it been really established the position of protein within EVs? And if so, do you think that some proteins, I mean, I know that I've seen some reports that, for example, for Alzheimer's disease, if EVs have a beta attached to the membrane, it will be some kind of a ticket to for EV to be uptaken much faster than other EV source types. Yeah.
So, so the prion protein has a GPI anchor, so that anchors it to the cell surface, and it actually maintains that anchor on the EV. So we did some studies where we took the EVs and there's this biochemical trick where you can use a detergent called Triton X114, and that will separate out GPI anchored proteins into one fraction and other proteins in the other. And we took the EVs and did that and showed that the prion protein still had its GPI anchor attached to the membrane when it was in an EV. So we know the PRP is on the surface of the EV. With respect to A beta, that is a transmembrane, that's when that's been cleaved from APP, it's embedded in the membrane and it remains there in EVs as well. Alpha-synuclein's a bit different because that's actually a secreted protein. It's one of these proteins that doesn't have a secretion sequence, but is found to be released. And we think that it finds itself into the cytosol of the EVs. And we think it also attaches to the membrane just in that stochastic method that I mentioned earlier. So the different proteins do have different subcellular localizations in the cell, but they do find their way into EVs for specific reasons, either being transmembrane, GPI anchored, or they're actually found within the cytosol of the EV.
Okay. I'm asking because in our research with A-beta, we find that A-beta mainly in the form of alpha helix, which is probably normal form for transmembrane proteins. And but I'm not sure. So for A-beta to be toxic or misfolded, it has to be in a beta sheet form, because there is no consensus, as from my knowledge in the research community, that A-beta has to be in the form of beta sheet or alpha helix. Both forms can be toxic, right?
Yeah. So, you know, there's the idea that soluble oligomers of A beta, so rather than the beta sheet rich plaques you find in the brain, but these soluble oligomers are more toxic than the plaque material. So, and A beta is a bit you know, it can exist in either an alpha helical conformation or that beta sheet amyloid form as well. So it looks to be that the pre-amyloid form, which isn't necessarily beta sheet, is more toxic than the actual amyloid form. And that comes back to, you know, protein misfolding diseases where the body could be sequestering these misfolded forms into amyloid as a way of stopping that toxic process. So it's you know, stopping that reactivity of the soluble forms, which are more toxic.
Yeah. Okay. Thank you so much.
So sorry, we're late, and there are several more questions that I think people can ask by email to Andy. I just wanted to take the last one from Michael Grainer, if he's still here.
But I can see his question on the chat, and that's it. You know, yeah, up to 60% of proteins are misfolded and then forced out into the cytosol for proteolysis. So, yeah, misfolding happens all the time. I think it's yeah, it's something that our cells just have to deal with. There's Michael.
Yeah, I'm on. Sorry, Mike. I couldn't get my mouse to work and unmute myself. So this is just something that's kind of known in the heat shock protein chaperone field, that quite a few proteins actually don't make it as they come off the ribosome. There's all sorts of ways to be misfolded. And the chaperones are try to salvage that, and when they can't, they will assist in getting rid of those things. And in particular, in the ER, this requires a whole batch of modifications that can very frequently fail and lead to these sorts of unfolded protein diseases as well. So I just want to point that out as a component. Yeah, protein folding is not always very successful, even though we love to think of it as a perfect system. So just threw that out there.
Okay. Okay, so I think we can close here. Thank Andy again for such a wonderful presentation.